Yeah, those tests are interesting. Very, very interesting. Alrighty. Okay. All right. Most of you are here. Let me go ahead and get GoGuardian started before I try to do anything else so that it doesn't block. All right, just a quick recap. We were doing mitosis. We're talking about cell division. And let me ask you a question. Where does cell division take place? Or I should say, where does cell division originate? Obviously, it takes place in the cell. But where does it originate? What is the central focus of mitosis? And I wanted to say sometimes y'all could unmute and it's faster than typing, especially if it's just a short answer. All righty, everybody's here today. Yay. Excellent, excellent. Maxwell, you're here, but no, he's not here. Trying to log in, but not logged in. Okay. He's got his computer on, but not in class yet. Getting there though. All righty. So where does nobody typed an answer in the chat? Where does cell division originate? That was a question. That was a question. There we go. All right, everybody's here. Nobody. Wake up, y'all. Thank you, Landon. In the nucleus, in the nucleus. All righty. So what we're looking at is what's happening in the nucleus that gets everything started. The cell cycle. Does mitosis happen all the time? Or is there something else that's happening during that cell cycle other than mitosis? Question for the chat. And again, if you want to unmute to answer, it's faster than typing. Thank you, Natalie. Interphase. And what's happening? There's three things happening during interphase. What's happening during interphase? So we got mitosis happens on a small window, and most of the time it's an interphase, but there's three things going on in interphase. Thank you, G1, S, and G2. So what's going on during G1, S, and G2? Okay, Natalie says G1, the cell is growing and it's preparing its DNA for replication. Okay, good. And the S is the replication. Good, thank you. And what's G2? Piper said the cell expands, the DNA is replicated and is checked. Um, G2 is preparation for, okay, growth in organelles. All right, so thank you, Natalie and Piper. So we have a period of time during interphase when the cell is getting ready to go through mitosis. So mitosis is the actual dealing with the replication and dividing the cell in two. But before that, the cell makes a copy of its chromosomes. And then, well, its cell grows. First of all, it grows, and then it makes a copy of its chromosomes. Humans have 42. So by the time they're finished, we have doubled the number of chromosomes, but they stay together. So what mitosis is going to do is rip those carbon copies apart. Sorry, 46 chromosomes, going to be 92 um, when they duplicate. And then the rest of the cell organelles have to divide up too, because when this cell eventually divides, you don't want to have empty and a new cell with empty organelles. So everything else in the cell has to duplicate as well. But the big part is that the chromosomes, and that's the big part because the chromosomes are the instructions. They tell everybody else what to do. They are the blueprints, the master design of how to create the next cell or the next organism. So the duplicating the DNA is extremely important, and it's extremely important that it does it correctly. So later on, we're going to talk about what happens when it reads it wrong, and we had talk having mutations. All right, let me finish saving the attendance, and then we'll share this Nearpod. Okay. All righty. So mitosis, where does mitosis happen? Mitosis is the type of cell division that happens where? Does it happen in all cells all the time? What kind of cells are going to undergo mitosis? That's an important question because you need to know that before we do meiosis. What kind of cells undergo mitosis? Somatic cells. What are somatic cells? Thank you, Landon and Zyla. What are somatic cells? What do we mean by somatic cells? Non-reproductive cells. That's good. All right, so know that vocabulary word. Thank you all for using that. Somatic cells are non-reproductive cells. That means all the other cells in your body, all the cells in your body, your eye, your ear, your nose, your arm, your leg, heart cells, those are all somatic cells. All right, so what we're going to look at today are the other cells that are not somatic cells, the reproductive cells. So I put the Nearpod link in the chat, and let's take a look at what meiosis is. Okay. Let's All right, give everybody a minute to get in. Oh, I'm gonna stop sharing. I forgot to, there's one little click that I forgot to do while sharing. So many little clicks to do. Okay. All right, so I've clicked on the sound button. Hopefully everybody's logged in. I'm gonna go ahead and start the video. And again, it's going to stop as Nearpod does for answers, interactive answers. So please be logged in and respond. Participation. I get a report who's responding and what your answers are. And so make sure that when it stops, you're there.
How did you end up with your dad's smile while your nose looks exactly like your mom's? Today, we'll be learning about the process of meiosis, from its many stages to the role it plays in determining our traits. Meiosis, unpacked.
Okay, so take a minute. It says, what do you already know about meiosis? So I'm going to hide that. So we just talked about what it might be. So tell me what you already know. We know it's not mitosis. All right, let's see what you got to say. Oh, so I'm looking at no answers here. What happened? Oh, thank you, Ariana. All right, and Piper. All right, Isabella. Okay, we're getting there. We're getting there. All right, so let's start from the bottom. Type of cell division. And folks, don't take the time to Google it and copy and paste. Oh, okay, Natalie says it reduces the number of chromosomes in half. All right, so now all of that vocabulary. So make sure that you're paying attention as we go along because that is the key part right there. It is, so it is, and as Ariana says, it is cell division for sexually reproducing organisms that reduces the number of chromosomes in the gametes. So gametes is a vocabulary word. Haploid cells is a vocabulary word. What else have you said? Um, Mitosis, if it does not work, there's birth defects. That's true. We're going to talk about um, mutations. Okay, replicates your DNA. Not exactly. It makes sure it's part of reproductive in the cell. All right, Kashawn and who else? Kashawn, right now you're the only one who hasn't answered my question. Okay. All right, everybody should have an answer in there. Thank you. Okay. All right. Thank you. Thank you. Thank you. So we can move on now.
Most cells clone themselves through mitosis, which replicates a cell with a complete set of 46 chromosomes, called a diploid, into two identical daughter cells.
All right. She said two things. She said they clone themselves. When you think of mitosis, think of cloning. They make an exact copy of what the parents look like. And that's important to remember. The other word down here is diploid. Okay. Diploid means they have two. Okay, create two identical daughter cells. So again, we have the parent cell and we have daughter cells. There's no son cells, they just call them daughter cells. So again, vocabulary, make sure that you are able to use the vocabulary.
But meiosis has to do with some very unique cells, or gametes, or sex cells. In males, these are sperm, and in females, eggs. Since offspring are produced when the two combine, if our gametes underwent mitosis, you'd end up with twice as many chromosomes as you actually need. Instead, sex cells reproduce through meiosis. During meiosis, a single specialized diploid cell divides twice to produce 4 cells containing half the original amount of the genetic material, so 23 chromosomes in total. The resulting cells are called haploids, and each one is genetically distinct from the others.
Okay, I'm going to switch that over. All right. What would happen if the egg, and folks, don't take the time to look it up. Write down, you were supposed to be listening. What would happen if the egg and sperm just underwent mitosis? Because technically they do both. So everybody needs to write an answer. Everybody, and don't look it up. These are designed to see if you're paying attention, not designed to see how fast you can Google the answer. All righty. All right, we got half of you answered the question correctly. The offspring would have double the number of necessary chromosomes. Okay, because the egg and the sperm are the ones that create the new individual. So if the egg and the sperm come together and each of them has 46 chromosomes, then the organism that they create will have 92 chromosomes, and we don't want that. Okay, so we want to reduce the number of chromosomes before the egg and the sperm meet.
Though meiosis differs from mitosis in a number of ways, it actually involves all the same stages, just repeated twice, in order to produce double the number of daughter cells. But before we get started, we have to go through interphase 1, during which the DNA and protein floating around in the cell's nucleus clump together to form chromosomes, then duplicate themselves, creating 2 copies of each strand. Every single chromosome then links up with its duplicate to form an X-shaped double chromosome. When 2 chromosomes are connected like this, each half is called a chromatid. Next comes the first real stage of meiosis, prophase 1. Each double chromosome lines up next to its homologue. In other words, your mother's and father's versions of the same chromosome. At this point, one chromatid from each double chromosome crosses over to the
Okay, she mentioned a vocabulary word again, homologs. Homologs mean that when you're looking at chromosomes, everybody has similar chromosomes that code for the same things. So when they're trying to, they map the chromosomes, they want to know this is the one, and they number them. This is the one that codes for all of these body parts, and the one from the sperm has the same chromosome for same characteristics. So certain chromosomes code for certain structures, or structures, okay? But they're the same. So what she's saying, homologs match up, they line up together so that we know the one from the egg is going to line up with the one from the sperm that are coding for the same characteristics.
the other and reattaches a portion of itself, trading sections of DNA. The swapped segments come from the same location on each chromosome. So one might be giving up its genes for, say, height, and getting the other chromosome's genetic code for that characteristic. Since the 4 chromatids swap sections of DNA at random, they're all different from one another.
All right, question. How is swapping of DNA, genetic material, related to heredity? Close that while you answer it. What is the advantage of swapping material? Okay. Come on, folks. Y'all, everybody's supposed to wake up, wake up. Everybody start answering. Got 2 answers. Everybody should answer. All right, got a couple. Okay, thank you, Ariana. That's a good answer. Oh, Natalie said it creates diversity. I like that word. All right. Okay. Now, we're Piper, in relation to your answer, and then I'm looking at someone else's answer as well. You're going to get traits from both anyway, because that's what when the egg meets the sperm, they're bringing their traits together. So you are always going to get traits from mom and traits from dad. What's going to happen is you're not going to do identical. It's like Ariana said, they will not be identical because we can shuffle it a little bit. And so then when we do the duplication, we're going to get a variation. Okay. How were doctors able to study? They they microscopes, and they have done a lot of testing. There is one, it's called the genome study. And I'm answering a question. Natalie asked a question in the chat. How were doctors able to study this to this degree? They took a long time. And if you want to look it up, it's called the Human Genome Study. They made a whole documentary of it. And if you put all of the information that they've discovered in this study, it filled up the file cabinets for, a whole classroom kind of thing. But they have done extensive research on mapping genes, and y'all are going to have to map genes in a little while. And actually looking how each section of the gene codes for a different trait. Like she said, one parent's tall, one is short, but the genes for tallness will line up, and the genes for, let me back that up. The gene for height will line up. So one parent could be tall, one parent could be short, but those genes will line up. And so later on, we're going to talk about what happens when they're different. Okay, what do we see when they're different? So we're going to talk about all of that. But they have done a lot of study on what each section of the gene looks like. And this is how we know about certain diseases and what causes them. Because they have done what they call genetic mapping. Good question. All righty.
The next 3 stages are a bit simpler. During metaphase 1, the newly reconstituted homologous chromosomes move to the center of the cell before anaphase 1 pulls them apart. Finally, with telophase 1, the double chromosomes begin to uncoil, and a new nuclear membrane forms around them. Then a crease called a cleavage develops between the 2 cells, and the 2 nuclei move apart from each other, eventually splitting apart in a process called cytokinesis. And that's the end of round 1 of meiosis. We now have 2 haploid cells made up of unique genetic combinations from the original chromosome pairs.
Okay, so let's answer this question, and I'm going to take that off so that you don't see the answer right away. Because what I notice is once you see my page, everybody changes their answer. So let's see what answers you give at the beginning. Which of the following are the correct sequence of the first round of meiosis? So again, remember PMAT.
At this point, we're only halfway through the process. We still have another 2 cells to make, so it's time for prophase 2.
Hold on, she didn't.
Made up of unique genetic combinations.
Okay, there we go. All right. All right, so again, PMAT is PMAT. So that's, and remember interphase, so that what makes D wrong is interphase is really not part of mitosis. So again, remember that it does mitosis first, and then it's going to do something different, but it's going to do the same stages. So prophase, metaphase, anaphase, telophase. So again, that, and then they have ones next to them because we're going to repeat these phases, but there's a phase 2. So, okay, so mitosis only has 1 phase, but meiosis or meiosis, depending on how you want to say it, has 2 phases. So we say prophase 1, metaphase 1, anaphase 1, telophase 1.
At this point, we're only halfway through the process. We still have another 2 cells to make, so it's time for prophase 2. First, the DNA and protein in the 2 daughter cells coil up to form double chromosomes, like the original cell did way back in interphase 1. Then, in metaphase 2, the double chromosomes move toward the middle of the cell, and in anaphase 2, they're pulled apart into individual chromosomes. And that brings us to the final step, telophase 2, which goes down exactly as it did in the first round, with the chromosomes drifting apart, then ultimately separating via cytokinesis. So, from one original cell with 46 chromosomes, we now have 4 new cells with 23 single chromosomes each. In males, each one is roughly the same size, with slightly different genetic information. But in females, the process results in just a single viable egg. Back in telophase 1, more of the original cell's features, like the cytoplasm and various organelles, are transferred into just one of the daughter cells. The same thing happens in telophase 2. So even though meiosis in females still results in 4 daughter cells, only one ends up with all of the other cellular stuff that it takes to make an embryo.
All right, so let's work on this question. For males, meiosis results in, and in females, it results in what? I think they want to know how many cells. All right. So it's males and then this might be a little confusing because like she said, both of them are going to create 4 cells, but with the sperm cells, they're going to actually be 4 identical cells, or not identical, but 4 distinct cells. And with the egg, they have one functional, she said, use the term viable, one functional cells, the other ones become discarded. Okay. So that's just there to be part of the reduction process, and then the other ones are discarded.
Let's recap. Meiosis is how our sex cells replicate themselves. It involves 8 phases and produces 4 cells, each of which contain half the amount of genetic information as the parent cell. And since that information is swapped randomly between chromosomes, it ensures that no 2 family members are exactly the same.
Okay. Question. What traits do you share with members of your family? What traits of yours are different? And this is a personal question. So you just meditate for a minute. You can't Google this answer. So this is about you. What traits do you share with your family members, and what trait do you have that is different? So you might say, I look like my mother, but I have a different feature. I might look like my brother or sister, but what's different about you? I give you another minute. All right, so let's look at some of your answers. Anchor issues for the same trait, different. Okay, I like that. Look like your mom, but talk like your dad. Share my hair and skin color, but different heights. Okay. All right, Malik, no answer. Maxwell, no answer. Let's get an answer up there by the time I finish looking at this. All right, Dad said my families, they need glasses. Ah, now I need, okay. Okay. Body shape and type, okay. And as you go, these are all traits, by the way, so that you all are pointing out traits. So that's good. Look like my grandma, but she had blue eyes. And we're going to talk about why somebody can have blue eyes in your family and somebody else have brown eyes. So we're going to look at that. Okay. Some like my parents, except for different facial features, freckles. So we're going to talk about freckles and we're going to talk about glasses. Okay. All righty. Good. All right, Malik, we're still waiting for you. Same height. Okay, thank you. You have the same hair color as who? Is everybody? Everybody in your family has the same hair color? All right. I'm going to move on though. I'm going to move on. Thank you for adding an answer. Okay. Let this finish. All right. So we're going to go back to Canvas now and look at meiosis in Canvas. So actually, let me just go ahead and make sure I end this one. All right. Thank you. Yes, Natalie, that is true. That is true. Brown is dominant over blue, and blue is recessive. So we are going to talk about dominant and recessive traits. And it's not that the gene is not there. Okay, so you're still getting the gene, one from mother, one from the father. It's just that one is dominant. So we are going to use those vocabulary. Thank you. Thank you for sharing. All righty. So yeah, so as we go through genetics, right now we're just talking about cell division, but we're going to move from cell division into genetics. And so those kinds of vocabulary words will come into play. All right, so let's look at this one. So again, what this is, well, they've answered the picture for you. But what you're looking at in this picture, like it says, one sperm cell with 23 chromosomes, which is half the number. Remember, you got to remember those numbers. 46 is normal for humans. And a gamete, so these are gametes. Egg cell is female gamete, and sperm cell is male gamete. So you need to know those vocabulary words. So when somebody asks you what is the male gamete called, that is the sperm cell. And so the sperm cell with its 23 fertilizes, it penetrates the egg cell, and then the chromosomes reunite. Okay. And so we call that word, and we have that next word, fertilization. Okay, the process of meiosis and of fertilization let's see what this video is going to tell us, account for creating a new, it restores the proper number of chromosomes. So by fertilization, we get the 46 chromosomes back.
Meiosis notes. Cell division of the sex cells, sperm and egg. Before we start, number 1, mitosis is the reproduction of all cells except sex cells. Number 2, gametes is another name for sex cells. Number 3, meiosis is the reproduction of those gametes. And number 4, the cells that result after meiosis have half the number of chromosomes. The reduction division. Meiosis is known as the reduction division because the chromosome number reduces by half. The cell divides twice in meiosis.
All right, I'm going to pause it there. So you should be taking notes. All right, so you need to write down that meiosis is known as reduction division because the chromosome number is reduced by half. Now, we keep talking about humans and 46 chromosomes with humans. This is happening all living things. So if you were an onion cell with 12 chromosomes, we're going to reduce this number by half. It'll be 6. So this is the reduction of all gametes in all living things, but they're all going to have a different number. But it's just easier for us to focus on humans because that's who we are. Okay. All right, so the second thing that's important to remember about meiosis is the cell divides twice. So at the end of mitosis, you ended up with 2 identical cells. During the first phase, you're still going to end up with 2 identical cells, except they're going to have half the number of chromosomes and not the restored number like before. Okay. Now I'm going to go ahead and play this. So if you're not done, you can go back in your Canvas and replay it as you should and complete anything that you need to complete.
Meiosis 1 and meiosis 2. This results in 4 new cells, each with half the number of chromosomes. Take a minute to look at these keywords. Remember what sister chromatids are. Review what a tetrad is. Remember what homologous chromosomes are. Look at diploid cells and haploid cells.
All right, I'm going to pause it there because we need, like I said, vocabulary, vocabulary, vocabulary. All right, so 2 identical chromatids are called sister chromatids, and together they are called a chromosome. So when we have, like they said, this X-looking structure, it's X-looking structure because one chromatid is attached to the other chromatid that is its carbon copy, and they're held together in the middle by a centromere, and together they are called a chromosome. Okay. All right. Tetrad. In interphase, the chromosomes make a copy of themselves. This results in 2 pairs of chromosomes or 4 chromatids. Okay, so again, where you had what looked like X and X, they make a copy of themselves, and now you've got 2 X and X's. Okay, which are 4 chromatids held together in the middle. So it looks, and then we say that there are 2 pairs. There's four chromosomes, but 2 pairs because they're linked in the middle, so they're pairs. All right, remember that homologous chromosome is a pair of chromosomes. Not only is it a pair of chromosomes, they're a pair of like chromosomes. They are coding for the same characteristic. And we'll look at that again later when we do genetic mapping. Right now, we're just talking about cell division, but you also need to know that like chromosomes are homologous pairs. All right, 1 copy comes from the mother, 1 copy comes from the father. Diploid cells have 2 of every type of chromosome, 1 set that came from each parent. Haploid cells have 1, so that's mitosis. Haploid cells have 1 of each kind of chromosome, also known as sex cells or gametes. So gametes are haploid cells. At the end of cell division, so at the end of mitosis, you should have diploid cells. All the cells should have two of every kind of chromosome. At the end of meiosis, you have haploid cells, one of every kind of chromosome. And again, if you need to go back, as you should.
Remember we discussed that meiosis happens twice. There's two stages. The first stage has prophase, metaphase, anaphase, telophase, and cytokinesis. The second phase is very similar. Again, we will discuss this in greater detail later.
All right, so again, look at the pictures of these. The phase 1 and phase 2, the same activity is going on. So during prophase, we're still getting ready and the nuclear membrane is disintegrating. During metaphase, again, the chromosomes are lining up in both phases down the middle. During anaphase, in both phases, they're being pulled apart. The difference is the number of chromosomes. Okay, that's what the difference is with each of these. During telophase, they start to form nuclear membranes again around each set of chromosomes. And during cytokinesis, the cell actually divides and creates two new cells. The first phase, you're only creating two cells. And those two cells are now moving on to the second phase. So two cells are going to divide again. And that's why we have 4 cells. Okay, notice they're calling it N. So remember that they're going to use that N number a lot. And that's just like algebra, it's going to be generic. So when we talk about it, we're going to know what the N number is because it'll be for humans. But if we're talking about another species, it'll be a different number.
Why is meiosis even important? Well, number 1, it reduces the number of chromosomes for sexual reproduction. Number 2, it allows offspring to be produced that are genetically different from the parents. And number 3, the generic variation, or genetic differences, are introduced by crossing over, random arrangements of tetrads, and random combinations of sperm and egg.
Whoops. All right, pause it there. All right. In the other video, they called it swapping. In this one, we need to know that it's called crossing over. Okay, so when the genes line up and they swap genes, the vocabulary word for that is crossing over. And it does it randomly. It doesn't, it does a section. It's not going to take a middle piece, but it's the end pieces are going to swap places. Okay, so know this word crossing over, or the term crossing over is when the swapping of genetic material. And it's random. There's no rhyme or reason to which one's going to do what.
Random combination of sperm and egg. In gametogenesis, there's oogenesis, which produces 4 cells, 1 large ovum, and 3 tiny polar bodies. Only the ovum will have a chance to survive it being fertilized. In spermatogenesis, this is where meiosis produces sperm, where all 4 are tiny and all 4 are capable.
All right, more vocabulary. So you have oogenesis. So think of eggs when you see the 2 O's. And those are going to create the 4 cells. But of the 4, you're going to have 1 large and 3 tiny, what they call polar cells. The polar cells are going to be discarded. They're going to eventually disintegrate. Only the ovum. So you got a lot of vocabulary here. Oogenesis is for egg cells, the creation of egg cells, and you're only going to get 1 large ovum. And then the spermatogenesis, creation of sperm cells, is going to create 4 equal size cells that eventually will get a tail so that they can move. Okay.
Take a minute to take a closer look at this picture of gametogenesis.
All right, I want you to draw that. All right, so take a minute and draw this. So you want a title, gametogenesis. So that means the making of gametes. And that should be an O. It kind of looks like a C up there, but it should be primary oocytes, OOCYTE, and primary spermatocytes. Okay. All right, so you have these initial cells that are going to divide to create two. And notice by this drawing of the secondary, and again, that should be an O. So it kind of looks like a C again, but it's oocytes, OOCYTE. But notice that one of those is already starting to look a little bigger than the other one. All right, so that's our first stage of my meiosis is the primary stage where they divided and created two cells. Now these cells don't have carbon copies. They have the regular number, but they don't have carbon copies. All right, then we have it says mature ovum is going to be the larger cell. So with the oocytes, make sure that you draw, again, one larger than the other and three kind of small ones. They're called the small ones are called polar bodies. And they just kind of go away. Whereas with the sperm, all the cells are going to look, all the secondary and spermatids are going to look the same size. All right, so this is kind of a graphic organizer to give you a visual of you start with one cell, the one cell makes two cells, and those two cells make four cells. Remember that the oocytes are the females and the spermatocytes are the males. All right, and just put a thumbs up on the screen if you're finished drawing. In your reactions, put a thumbs up on the thing so I'll know when everybody's done. All right, I didn't get a thumbs up from everybody. So remember, when you're done, let me know. All right, so I'm gonna give it another minute for you to finish, and then I'm gonna go on. And again, this is in your canvas, so you can go back and finish drawing if you need more time to do that. Okay. All right. So again, with these, you can click on each of these and read what is happening in each one. So again, here they use the term crossing over, where they exchange genetic material. Now, this only happens in meiosis. Mitosis does not have the crossing over. With mitosis, the prophase the chromosomes are exactly like the cell that they started with. There is no crossing over. So there'll be questions that ask you, what is the difference between, some differences between mitosis and meiosis? There is no crossing over in mitosis. All right. This one will be the same. Again, remember the term homologous pairs. I think, and then telophase is where after all of the chromosomes have separated them, they have to be reorganized back into the nucleus. All right. And again, the big difference is phase one creates 2 cells and phase two creates 4 daughter cells that have half the number. So the first phase, they still have the same number, but they have half the number of chromosomes by the time you get to phase two. All right, so I'm not going to repeat all that. And this one, while she's a little bit too enthusiastic for me. Okay, so I'm going to come down here and I want you to click on this and get started with this quiz. Okay. All right, so what did you say? If a haploid number is 10, what's the diploid number? Good. All right. Next one. Which of the following describes the products? Products means like, what do you get? What do you get after the process is all over? All right, somebody else share with me in the chat. What's the correct answer for this? You're going to get 4 haploid cells. Correct. All right. Oogenesis produces how many viable cells? Okay, and spermatogenesis, okay. So it's going to be 1 and 4. 1 viable cell and 4 sperm cells. All right, during meiosis, where do you see the crossing over? Prophase 1, yes indeed. Okay. All right, not prophase 2. So prophase 2, they're still going to be replicating, reducing and organizing like the other prophase. All right, which of the following best describes a tetrad? Now this is practice quiz. I'm going to give this as a real quiz for a grade. So hopefully you're paying attention. Okay, homologous chromosomes. Is that right? Okay. All righty. Homologous pairs separated meiosis. Which one? And sister chromatids separation and mitosis what? All right, 1 and 2. Okay. Thank you, Zyla. And Natalie. Alrighty. In this phase, the nuclear membrane reappears and there is a brief resting period before the next one. What do we call the resting phase? Well, there's something going on during telophase. So what is always the resting phase? Yeah, okay, let's come back up here. Let's look at this picture. All right, this is telophase 1 and 2. It's not exactly resting during here. Okay, so what is our resting stage always? The answer is telophase 1. Okay, interesting. It should have been interphase. There is an interphase. There's a resting period. All right, non-disjunction. Yeah, I was looking at this number 7 up here. So look up here at number 7 in our section up here. Non-disjunction. So if we don't do this properly, what is going to happen? What's going to happen if if the chromosomes don't reduce in numbers? All right, it will be genetic disorders, but it's actually, so you're going to have, they don't separate properly, occurs when they don't separate properly. So that's what it says up here. If they don't separate properly, it can cause some of the genetic disorders when you have maybe too many eggs or too many sperms, but it's all of the above. It should be all of the above. Okay, why is mine not clicking? I guess that's it. Okay. Check mark. Okay. Okay. Thank you. All righty. So make sure that you any questions about those. So again, what you really need to do is make sure that you go through this, ready to answer those questions. And then before you go, what you need to know. So you need to understand, why do we have meiosis? We need to reduce the number of chromosomes so that when the egg meets the sperm, they are reuniting the original number. They're not bringing 46 and 46 to create the new organism. They're bringing 23 and 23 to create the 46. Okay. What are the two differences? Again, you need to know what's the difference between mitosis and meiosis. So mitosis has one stage, meiosis has two. Mitosis has two cells that are produced, meiosis has four. Okay. The other one is mitosis has diploid numbers. It's going to have the 46. So for humans, 46 is our diploid number and 23 is a haploid, homologous pairs. So in other words, if you pair up your 46, you have 23 pairs. Whereas in meiosis, the haploid cells, which are the gametes only, have one kind of chromosome, and each cell only has 23. They don't have two. They don't have homologous pairs. They have 23. All right, any questions about that? No? All right. So like I said, when I see you next, I might have a little quiz for you. So ask questions if you need to now. And again, look over this again. I'm trying to make sure we have access to the program called Gizmo Labs so that we can get some labs. Okay. All right. Well, if you're good, then I'm good. And you all have a nice weekend.